Dopamine, the function, meaning, and side effects of neurotransmitters. Dopamine, addiction, connection, and ADHD issues. Chapter 1. What is dopamine? What is the definition of dopamine? What does it do and in what way? These kinds of questions have caused debate in neuroscience for a long time. A new study from the UK could have some of the answers. The word dopamine means very different things to different people. From drug addiction to Parkinson's illness to a Hollywood film, dopamine is a part of mainstream culture and also an enduringly interesting research subject in neuroscience. It has been part of over 110,000 research documents in the last 60 years, but is still a source of controversy among neuroscientists. Attempting to sum up the function of dopamine in a brief chapter or book isn't going to be simple. I am going to leave a lot of researchers dissatisfied and some totally mad. Let's start with the basics. Dopamine is a neurotransmitter, one of those chemicals that is accountable for transferring signals between the afferent neurons of the brain. Really few nerve cells actually make dopamine. Some, in a part of the brain called the substantia nigra, are the cells that die during Parkinson's disease. The functions of others, situated in a part of the brain called the forward tegmental area, VTA, are less well defined and are the major source of the previously mentioned debate, which is my entire focus as well. When dopamine neurons end up being activated, they release dopamine. One of the best described roles for VTA dopamine nerve cells is in learning more about benefits. VTA dopamine neurons become triggered when something good happens all of a sudden, such as the unexpected accessibility of food. Most mistreated drugs cause the release of dopamine, and this is thought to add to their addicting properties. However, what about bad things? Do they trigger dopamine nerve cells? It's maybe even more crucial to learn when something bad is going to happen than when something good is. With predators or illness, you typically do not get a second chance. Is dopamine associated with learning more about bad things? Herein lies some of the debate surrounding dopamine. Not all the nerve cells in the VTA make dopamine. Most research studies had suggested that the unexpected discussion of aversive or noxious stimuli like pain triggered the activation of some nerve cells in the forward tegmental area. But were these dopamine nerve cells? In 2004, Mark Unglis and co-workers at the University of Oxford published a paper in the diary Science suggesting that dopamine nerve cells were widely inhibited by aversive events. They used a painstakingly detailed approach to determine those nerve cells that were triggered or prevented by aversive stimuli, and after that biochemically evaluated those neurons to determine if they truly were dopamine neurons. They did find that some nerve cells became triggered by aversive stimuli, but these neurons did not make dopamine. The findings were really clear, but were controversial. They did not agree with other research studies on the role of dopamine, which include some which revealed that treatment with drugs that blocked the action of dopamine could block discovering aversive events. Moreover, chemical measurements showed that dopamine was released by animals undergoing a difficult experience. If dopamine neurons are not triggered when finding out about aversive events, how is this dopamine being released? And why would blocking the impacts of dopamine prevent learning about aversive events? Unglis and Company, now at the United Kingdom Medical Science Research Study Council, Imperial College London, hypothesized that the devil may be in the detail. Maybe the VTA is not a single uniform part of the brain, but is made up of functionally different subregions. Before performing their newest study, published in the Proceedings of the National Academy of Sciences, they returned and looked again at the literature about dopamine nerve cells in the forward tegmental area. They observed an experimental quirk. Most research studies of VTA dopamine neurons, those demonstrating that dopamine nerve cells aren't triggered by aversive stimuli, we're measuring reactions from the same small part of the VTA, called the dorsal VTA. What if neurons in a very different part of the VTA ended up being activated by aversive stimuli and released dopamine? To test this, they gave rats an electrical shock. Not an electrocution, just a moderate electric shock, much the same as a dog would receive from a shock training collar. During this electrical shock, they noted the activity of nerve cells from the dorsal VTA and a slightly different part of the VTA called the forward VTA. They found, like others had formerly, that nerve cells in the dorsal VTA were either inhibited by an aversive stimulus or didn't respond at all. By contrast, those neurons in the ventral VTA ended up being triggered by the shock. Truth be told, they became really activated, displaying a type of response referred to as, quote, burst firing, that would be expected to produce an extensive dopamine release. So that would appear to be that issue fixed. But there was another twist. As I wrote earlier, one of dopamine's best-known roles is in learning about benefits. Relief from an aversive event can be considered as a reward. The pattern of learning shown by animals and human beings is comparable when comparing a real reward, for example food, or relief from an aversive stimulus, for example when an electrical shock is shut off. What Unglis and company observed was that those nerve cells in the dorsal VTA, those that were unresponsive or were hindered by the electric shock, became transiently activated when the shock was shut off. That would be consistent with their role in reward learning, while also partially explaining why dopamine blockers impair discovering aversive events. So the story of dopamine has progressed a bit further while becoming a bit more complex. Not all parts of the ventral tegmental area are the exact same. Chapter 2. Giving Up. A new research study in mice suggests that dopamine-suppressing nociception nerve cells in the brain's reward and motivation center, called the ventral tegmental area, end up being really active right before a mouse reaches a breakpoint when it gives up on putting in effort to receive a sugary reward. According to the authors, nociception is, quote, a complex particle that suppresses dopamine, a chemical largely related to motivation. This paper, a paranegral VTA nociception circuit that constrains inspiration for reward, was published July 25th in the diary Cell. All of us know the deflated feeling of seeing that a dream or objective you've been pursuing is out of reach and unattainable. Usually, giving up is joined by a little voice in your head that says, I can't do this anymore. I need to quit. I forfeit. Even if you have been standing firm and struggling towards accomplishing a certain objective, like a rock climber attempting to reach the summit of a mountain. Within a millisecond, it can feel like the urge to give up becomes so overwhelming that it stops you dead in your tracks and forces you to throw in the towel. As an ultra-endurance athlete, I know from experience that the sudden desire to give up often appears to come out of nowhere. To avoid allowing this phenomenon to stop me from reaching the finish line and having a DNF did not end up beside my name on the results board, I thought of some road-tested approaches for bypassing the possibly immobilizing urge to forfeit throughout a race. Though previously, remarkably little has been known about the neural circuits that appear to block the brain's inspiration to look for rewards and the neuroscience of quitting. The recent discovery of a possible link between dopamine-suppressing nerve cells and giving up came to light via lab experiments in which mice needed to poke their snouts against a dispenser to get a hit of sucrose. Initially, one nose poke gave some sucrose, then it took 2 nose pokes, then 5, and so forth. The scientists purposely increased the amount of nose poking needed for a mouse to get the sucrose benefit. Using modern strategies, the scientists had the ability to recognize that just before a mouse reached the breakpoint of giving up, dopamine-suppressing nociception nerve cells connected to the VTA became very active. The creative representation beneath of this study by Max Huffman brings this research to life in a spirited and whimsical manner in which makes it simple to imagine what it feels like when nociception suppresses free-flowing dopamine supplies, constrains motivation, and increases the chances of giving up. In my mind's eye, the Huffman illustration illustrates dopamine as red rock climbing up bodies that are scaling a mountain, while the green tentacles of dopamine-suppressing nociception nerve cells are clenching their bodies in an effort to push them to the breakpoint of quitting their summit effort. Quote, we are taking an entirely new angle on an area of the brain called VTA, forward tegmental area. Co-lead author Christian Peterson, a fourth-year PhD student in bioengineering at the University of Washington School of Medicine and the UW College of Engineering, said in a declaration. For this research study, Peterson and co-workers spent 4 years analyzing the role nociception plays in regulating reward-driven inspiration and quitting. As pointed out, one significant emphasis of their research is that nociception nerve cells project in your area on VTA dopamine nerve cells and end up being extremely active when mice end up being demotivated to seek rewards. At the time of enjoyable and gratifying activities or pursuits, specific neurons in the VTA release dopamine. Previous research on inspiration and benefit has focused primarily on the activation of dopamine neurons, but neglected the dopamine-suppressing role of the nociception modulatory system. According to the UW scientists, their newest nociception-related discoveries could cause interventions that improve motivation in scientifically depressed patients experiencing debilitating demotivation. Targeting nociception neurons in different ways could also be used to curb the motivation to seek drugs or alcohol in those with substance use disorders. Quote, we may think of different situations where people aren't inspired, like depression, and block these neurons and receptors to help them feel better, senior writer Michael Bruchas said in a declaration. Quote, that's what's powerful about discovering these cells. Neuropsychiatric diseases that affect motivation could be enhanced. Seeking to the future, these neurons could perhaps be customized in people seeking drugs or those that have other addictions. Bruchas is a teacher of anesthesiology, pain medication, and pharmacology at the University of Washington School of Medicine. He's also a principal faculty member at the UW Center of Excellence in Neurobiology of Dependency, Discomfort, and Emotion, NAEP. From an evolutionary perspective, the scientists speculate that neural circuits linked to motivation and benefit in mammals are developed to protect energy when resources are limited. Because of this, nociception nerve cells could be a natural mechanism developed to force people and animals to cut losses, as opposed to continuing unsuccessful efforts to pursue a benefit because of sunk expenses. Quote, perseverance in looking for unsure benefits can be unfavorable due to risky exposure to predators or from energy expense, the scientists noted in a press release. The researchers are positive that one day their findings on nociception activation and quitting could be used to increase motivation in healthy ways and as an intervention to curtail reward-seeking behaviors associated with dependency. Is the brain wired to give up when a reward runs out of reach? Few things crush the spirit like the sensation of quitting. All of us know the gut feeling. When you thought an objective was in reach, then understand you just do not have enough of whatever it requires to arrive. But what if you learnt that this feeling of depletion is actually just another thing our brain does to keep its chemical symphony in tune? That's the conclusion of a new research study in mice focused on how the brain musters motivation in pursuit of rewards, and the other hand when the benefit is out of reach. Neuroscience already has a pretty good deal with on what happens when we're excited about pursuing a benefit, whether it's something tangible like cash, food, or sex, or more abstract like love or power, a similar chemical pattern plays out in the brain. The neurotransmitter dopamine floods neural paths in what's usually called the brain's benefit center. This is the biochemical dynamic that drives us forward, and it's no exaggeration to say that it's central to why we go after anything at all. But brains are instruments of balance, and as it turns out, mammals have another system that puts in a limiting force on the benefit rise, called the Nociception modulatory system, which also happens to be key to how the brain regulates pain. The nerve cells in this system, dubbed aggravation nerve cells, emit particles called nociception that reduce dopamine. In influence, nociception is anti-dopamine. Scientists found how this works by observing mice looking for sugar tucked away in a little port. To get the sugar, they had to poke their snout in and lick. The researchers made it easy initially to stimulate more motivation to get the products, but with each attempt, they made it a little harder for the mice to prosper. After making it so hard that the mice poked their snouts over and over and still could not get a taste, they finally began giving up. Eventually, all of the mice stopped trying. While this was going on, the researchers were tracking the rodent's neural activity and learned that nociception nerve cells were most active when the mice gave up. Not coincidentally, these neurons lie near the brain's forward tegmental area, VTA, the center of activity in the reward center, and the distance provides easy access to tap the brakes. Quote, the big discovery is that big, complicated neurotransmitters referred to as neuropeptides have a really robust influence on animal behavior by acting on the VTA, said co-lead author Christian Peterson, a PhD student in bioengineering at the University of Washington School of Medication, in a press statement. The reason for this interplay between reward surge and reward suppression boils down to the brain's tendency towards stability and balance, aka homeostasis. The brains of mammals have integrated mechanisms to keep reward looking from going too far in either direction. In the wild, continuing to take part in risky reward-seeking behavior when success is out of reach could lead to injury or death. So we have very likely acquired this chemical balancing to serve as an evolved survival system. Conditions like depression and dependency may develop from these regulative systems not working well for any number of reasons. The scientists think the most recent research study could shed light on those and other conditions and maybe ultimately result in the development of new chemical interventions to help restore balance. That will require further replication in humans, of course, but this research is a strong starting point. Quote, we may think of different situations where people aren't inspired, like depression, and block these neurons and receptors to help them feel better, said senior study author Michael Bruchus, professor of anesthesiology, pain medication, and pharmacology at the University of Washington School of Medication. Quote, that's what's powerful about discovering these cells. Neuropsychiatric diseases that affect motivation could be enhanced. For now, the value of the discovery could be more straightforward in just understanding that quitting, after trying hard, isn't a character defect or moral failure. It's just another way that the brain keeps things level. Chapter 3. Motivation to exercise. Chronically non-active mice who eat an unhealthy diet plan and become obese display very little inspiration to move their bodies, according to a new report from the National Institute of Health, NIH, released online yesterday in the diary Cell Metabolic Process. Surprisingly, the researchers believe that the lack of inspiration to move stems primarily from flexible alterations in D2 type D2R dopamine-binding receptors in the striatum of the basal ganglia, not simply from being overweight or lacking willpower. Although this is an animal research study, these groundbreaking findings are a follow-up to similar human research carried out earlier this year at the NIH. The initial research study recognized a correlative association between problems of dopaminergic pathways, which deliver the benefit molecule dopamine to receptors in the striatum, and chronic inactivity. Dopamine receptors in the striatum trigger feelings of benefit that make your brain say, "Do it more." This helps to hardwire both positive lifestyle behaviors that end up being routines and also harmful addictions. In previous research studies, breakdowns of the basal ganglia have been associated with Parkinson's and Huntington's illnesses, autism spectrum disorders, ASD, obsessive-compulsive disorder, OCD, Tourette's syndrome, and other neuropsychiatric disorders. Did you make a brand new Year's resolution to sit less, exercise more, and eat better? If you're like numerous millions of people around the globe, odds are that you'll make a New Year's resolution to adopt much healthier lifestyle routines in 2017. Regrettably, the chances of sticking with your exercise resolution long enough for moderate to vigorous exercise, MVPA, to end up being a day-to-day routine are statistically slim. But there's hope. Simply because of the neuroplasticity of your basal ganglia, the NIH scientists learned that dopamine receptors are flexible and can be reshaped and rewired if you can stick to slight enhancements to diet and exercise long enough to produce an upward spiral within the striatal loop circuit. This improves dopamine D2R binding and makes perspiring feel good, which will make you want to work out more. Till recently, most experts would blame an absence of self-discipline as being the prime culprit for people failing to stay motivated to stick with an exercise program. Now, for the first time, it seems that problems within someone's D2R dopamine signaling, rooted in the striatum of the basal ganglia, may clarify why some of us are more susceptible to be lazy bones than others. All animals, including human beings, seek pleasure and stay away from pain. Without proper dopamine signaling, it is unrealistic for exercise to feel like a pleasurable or satisfying experience. If your D2 type dopamine receptors are inefficient, it can make exercise a really disagreeable experience on a neurobiological level. While exercise produces runner's high for some, this research helps to clarify why the link between exercise and feeling good isn't a universal phenomenon in a way that exceeds endorphins or endocannabinoids. In a statement to Cell Press, the study's senior writer Alexei V. Kravitz, a private investigator in the eating and addiction area, diabetes, endocrinology, and obesity branch at the NIH, said, quote, "In many cases, self-discipline is invoked as a way to customize conduct. However, if we don't understand the underlying physical basis for that behavior, it's hard to say that willpower alone can resolve it. We know that physical activity is linked to overall health, but not much is known a lot about why people or animals with weight problems are less active. There is a typical belief that obese animals do not move as much simply because carrying additional body weight is physically disabling. Having said that, our discovery suggests that assumption doesn't clarify the entire story. Other research studies have connected dopamine signaling problems to obesity, but most of them have taken a look at benefit processing, how animals feel when they eat different foods. We looked at something easier. Dopamine is critical for movement, and obesity is related to a lack of movement. Kravitz's other research fixates on basal ganglia circuits and how their function changes as a result of impacts such as weight problems, addiction, depression, and Parkinson's disease. When he started carrying out weight problems research several years ago, Kravitz was amazed to find comparable patterns of movement behavior between obese mice and lab mice with Parkinson's illness. Based upon this serendipitous observation, he assumed that the reason that both overweight and Parkinsonian mice were inactive was because of dysfunction in their dopamine systems. After evaluating 6 different parts in the dopamine signaling pathways in lab animals, Kravitz and co-workers pinpointed that inactive overweight mice had deficits in the D2 type dopamine receptor, but not the D1 type receptor. Although other elements are probably involved in this process, the deficit in D2R is sufficient to clarify the absence of activity in overweight mice, the researchers concluded. Striosome dendron bouquets in the basal ganglia may drive voluntary movement. The most recent findings by Kravitz et al. On D2 type dopamine receptors and inactivity dovetail with research being performed by MIT neuroscientists who pinpointed dopamine receptors in the striatum as being associated with decisions requiring a cost-benefit analysis. The MIT scientist used a revolutionary method developed at the Massachusetts Institute of Technology known as growth microscopy to focus on a direct communication pathway to the striatum that is directly linked to another complex dopamine-fueled subsystem in the basal ganglia. The researchers coined this subsystem a striosome dendron arrangement. The cluster of nerve cells in this bouquet are associated with making choices that require any kind of cost-benefit analysis, in which you weigh the pros and cons of a possible choice using practical rationale, combined with a gut feeling of the emotional toll connected to a specific choice. The study was led by Anne Graybiel, an institute professor at MIT and member of the McGovern Institute for Brain Research Study. In a declaration to MIT, Graybiel said that the striosome dendron arrangement might also be a prospective target for treating the neural degeneration seen in Parkinson's disease. Please take a few minutes to watch this YouTube video of Anne Graybiel describing her research on the basal ganglia at MIT. If you stick with it, habitual way of life choices can restart your dopamine receptors. The most recent discoveries on the capability of D2 type dopamine receptors to increase or reduce a person's motivation to move his or her body provide a neuroscience-based clarification as to why it's an uphill struggle for some people to psychologically kickstart their motivation to be more physically active through large force of will. Hopefully, this new empirical evidence will lower moral judgments and any stigma people who might be overweight and keep from exercise experience. The clinical proof suggests that a lack of inspiration to exercise has deep neurobiological roots and goes far past just being lazy. That said, the best news of the latest research from the NIH is that the dysfunction of the D2 type dopamine receptors isn't set in stone. By gradually increasing physical activity, eating a healthier diet, and losing some weight, the basal ganglia seems to reboot and dopamine receptors wake up. This kickstarts the biological reward mechanism that can make someone want to move his or her body voluntarily. Future research by Kravitz and his team at the NIH will concentrate on how eating an unhealthy, obesogenic diet influences dopamine signaling. The scientists also prepared to examine how long it takes for overweight mice to restore higher inspiration levels and end up being active once they begin eating a healthier diet, moving more, and losing weight. Chapter 4: ADHD and Dopamine. For the past some decades, scientists have been examining possible connections between ADHD and one of the primary chemical messengers in the brain, dopamine. Scientists noticed that medications that work for ADHD all lead to increases in dopamine at connections between brain cells. The most basic explanation seemed to be that the medications work by helping to fix a broken dopamine system in the brains of ADHDers. When it comes to biology, however, the easiest explanation isn't always right, and the issue is not so easy to settle. One of us, Ben, is a brain scientist. So let's talk a little about what's actually understood and not known about dopamine and ADHD. Dopamine is a neurotransmitter, a chemical that transfers signals between specific brain cells, nerve cells, at their connections, synapses. It is just one of several significant neurotransmitters in the brain, not only in humans, in the brains of all animals. Its chemical structure is very closely related to another neurotransmitter called norepinephrine, which is closely associated with the hormonal agent adrenaline, also called epinephrine. But the impacts of dopamine are quite different. Most or all of the drugs currently used to deal with ADHD do indeed increase the efficiency of dopamine at synapses. Something about this helps patients with ADHD focus and pay better attention to one thing at a time. How precisely more dopamine translates into better concentration and focus is not yet comprehended. Individually, there is also some genetic proof supporting a crucial role for dopamine in ADHD. ADHD has a considerable heritable part, and at the very least a few of the genes included encode dopamine receptors. These are the particles at your synapses that pick up dopamine signals. But it is definitely not simply a matter of too little dopamine or dopamine signaling in the brains of ADHDers. Dopamine's role in the brain is far more different and complex for this to be the case. This is an example. Not only is dopamine involved in focus and concentration, it is also a major player in the reward circuitry of the brain, and for this reason is necessary in all forms of dependency. It plays a vital role in a circuit involved in the production of the female nursing hormonal agent prolactin. It is also critical in the brain circuitry controlling smooth muscle movements that gets damaged in Parkinson's disease. Muscle rigidness and irregular jerking movements are one effect of too little dopamine in that illness. Not only ADHD medications, but other psychiatric drugs also affect dopamine. Antipsychotic medications modify dopamine signaling in approximately the opposite direction of the ADHD medications. Antipsychotics used to deal with hallucinations and messy thinking in mental conditions like schizophrenia decrease the efficiency of dopamine at synapses everywhere in the brain. The blockade of dopamine in some of the circuits we just discussed can trigger unwanted bad side effects from these medications, like muscle rigidness or milk production, even in men. While the blockade of the signal in other brain areas, like the cerebral cortex, the outer layers of your brain, appears to be handy in minimizing hallucinations and disordered thinking. To make matters even more complicated, each person has their own particular genetic variation in receptors for dopamine and other neurotransmitters. Not only is the amount of dopamine crucial, but the many different types of receptor present in unique regions of your brain, and the exceptionally complex ways these regions are all wired together, add to how you react to levels of dopamine and other neurotransmitters in your brain, and to medications that modify this balance. It would be super cool if your medical professional could scan your brain or your DNA and see your levels of dopamine, all your other neurotransmitters, the mix of signifying molecules inside your 100 billion brain cells, the kinds of receptors present at your 100 trillion synapses, the complex circuitry that wires all this together, and based upon that, anticipate how you'll respond to a medication. Nonetheless, at this point, that's pure science fiction. There is no scan available today that can expose in sufficient resolution your neurotransmitter levels, receptor subtypes, and the myriad other elements underlying the functional anatomy of ADHD, or addiction, or Parkinson's illness, or schizophrenia. And even if we had the innovation to perform such a scan, we would have not a single clue how to translate the results. As the saying goes, that day could come, but today isn't yet that day. So take the hype about dopamine and other neurotransmitters with a huge grain of salt. A little knowledge can be a dangerous thing. The human brain is aptly referred to as the most intricate structure in the known universe. Consider that big complexity, it is safe to say that brain scientists presently comprehend much less than 1% of it. We're still just searching around the edges of beginning to understand how the brain is assembled, let alone how it creates your thoughts and conduct. A lot of what doctors think they know about disorders such as ADHD has been presumed, rightly or incorrectly, from how existing psychiatric drugs appear to work. Drug A works in disorder B and increases chemical C. For that reason, disorder B must include insufficient chemical C in the brain. That has to do with as good as it gets, and frankly that is not great. So beware of anyone telling and selling you dietary supplements to increase brain neurotransmitter levels, sets to expose other supposedly relevant elements of your brain chemistry, or for that matter, DNA scans to tell you what will work or not work in you or your loved ones. We're still in an age of educated trial and error when it comes to practically all psychopharmacology, though with continuing research this will change, slowly in the beginning, more quickly over time. We just spent a lot of space here telling you all the don'ts about dopamine. Don't believe that ADHD or any neuropsychiatric condition is as easy as too little dopamine in the brain. Do not waste your cash on vitamins or dietary supplements to increase dopamine or other neurotransmitter levels. Don't waste your time on slickly marketed pseudoscientific scans that haven't been authorized by the FDA and do not have proven predictive value. What can we tell you that is a do? Here's one do that we feel is highly supported by both clinical data and good sense. Do exercise routinely. In trustworthy scientific research studies, exercise has consistently been revealed to increase dopamine in important circuits within your brain. If you put a lot of rats on treadmills, and after that cut their brains open to measure dopamine levels, the rats on treadmills show similar dopamine boosts in certain brain circuits to a lot of rats who have been fed Ritalin. Translating that to the human ADHD patient, regular exercise can improve your signs in a way that is at least rather comparable to the manner in which ADHD medications work. And, needless to say, workout confers various kinds of other health benefits, so exercise each day for the good of your muscles, your skeleton, your heart, and your brain. Chapter 5. The reverse of addiction is connection. What triggers dependency? In a progressively commonly disseminated TED Talk titled, "Every Little Thing You Think You Learn About Addiction Is Wrong," British journalist Johann Hari talks about the available research into the underlying reasons for dependency and concludes, rather remarkably, that the opposite of dependency isn't sobriety, it's connection. His statement echoes a specific theme that I and many 21st century addiction specialists have embraced for many years. The dependency isn't about the enjoyable results of compounds, it's about the user's incapability to connect in healthy ways with other humans. To put it simply, addiction isn't a compound disorder, it's a social disorder. This obviously contrasts with what most folks believe about drug abuse. Generally speaking, people think that the pleasurable results of alcohol, cocaine, heroin, and so forth are the primary motorists of dependency. And why not? We know for certain that once ingested, these substances trigger the release of dopamine and several other pleasure-related neurochemicals into the brain. In other words, potentially addictive compounds make us feel good, and just because we like to feel good, we tend to go back for more. Therefore, the human propensity for dependency, or so it seems at first glance. Bolstering this belief is the fact that the majority of the early research and theories on the root causes of addiction are centered on the brain's enjoyment reaction, the previously mentioned dopamine rush. Even the National Institute on Substance Abuse at first embraced this view. Nonetheless, this long-held belief is inaccurate. If it wasn't, then everyone who ever took a sip of alcohol would end up being a raving drunk, and everyone who ever consumed an opiate, even through prescription, would wind up in a street shooting heroin. But that is not even close to what actually happens. In reality, only about 10% of the people who try a potentially addictive substance ultimately end up being addicted. The rest of the people either leave the substance completely or continue to enjoy it delicately or recreationally. Welcome to the rat park. Given the above, one questions what's actually happening with dependency. Clearly, there's more to the formula than just the dopamine satisfaction reaction. Surely the experience of enjoyment does play some role, simply because it opens the doorway to addiction. But it is clear, based upon the fact that most people do not become addicts, that gradually an individual's initial experience of enjoyment is not what triggers that individual to go back to an addictive substance again and again, compulsively, and to her or his detriment. This is a problem that researchers started to explore in the late 1970s and early 1980s. An example would be the following. Canadian psychologist Bruce Alexander looked at the results of research studies in which rats were placed in empty cages, alone, with 2 water bottles to select from, one with distilled water, the other with heroin-infused water. Those experiments showed that as time passed, these rats would consistently get hooked on and eventually overdose from heroin. So the researchers unsurprisingly concluded that the potential of severe satisfaction, in and of itself, is addictive. Case closed, right? Not for Alexander. He was bothered by the fact that the cages in which the rats were separated were little, without any potential for stimulation beyond the heroin. Alexander thought, of course they all got high. What else were they supposed to do? In reaction to this perceived shortcoming, Alexander created what we now call the rat park, a cage approximately 200 times larger than the typical isolation cage, with hamster wheels and multicolored balls to play with, lots of yummy food to eat, and areas for breeding and raising litters. And he put not 1 rat, but 20 rats of both genders into the cage. Then and only then did he mirror the old experiments, offering 1 bottle of pure water and 1 bottle of heroin water. And guess what? The rats disregarded the heroin. They were far more fascinated by normal common rat activities such as playing, fighting, eating, and mating. Essentially, with a bit of social stimulus and connection, dependency vanished. Heck, even rats who'd previously been isolated and sucking on the heroin water left it alone once they were introduced to the rat park. The human rat park. One of the reasons that rats are regularly used in mental experiments is that they're social creatures in tons of the exact same ways that humans are social beings. They need stimulation, company, play, drama, sex, and interaction to stay happy. People, though, add an additional layer to this formula. We need to be able to trust and to emotionally attach. This human need for trust and attachment was initially studied and developed as a psychological construct in the 1950s when John Bowlby tracked the reactions of small children when they were split up from their parents. In a nutshell, he found that infants, young kids, and kids have a comprehensive need for safe and trusted caregivers. If kids have that, they have the tendency to be happy in youth and well-adjusted, mentally healthy later in life. If kids do not have that, it's a very different story. It is clear from Bowlby's work and the work of later scientists that the level and caliber of trust and connection experienced in early youth brings forth into adulthood. Those who experience secure attachment as infants, young kids, and little kids almost always carry that with them into adulthood, and they are naturally able to trust and connect in healthy ways. Meanwhile, those who do not experience protected early life attachment tend to have issues with trust and connection later in life. In other words, firmly attached individuals tend to feel comfortable in and to enjoy the human rat park, while insecurely attached people generally struggle to fit in and connect. Guess which group is more susceptible to addiction? The good news is that those with insecure attachment styles aren't locked into this method for life. With appropriate assistance and a fair amount of mindful effort, the individuals who were not enhanced with protected attachment in childhood, and for this reason, the capability to quickly connect in adulthood, can learn to firmly attach, typically through therapy, support system, and various other healthy and healing relationships, creating in time what's referred to as earned security. For the addicted, earned security is a really crucial principle. Rats don't actually need it. You can take an addicted rat and toss him into the rat park and he will quickly and easily assimilate, pushing his dependency to the curb in favor of healthier rat connections and activities. But people, not so much. With the addicted, there is further work to be done, and part of that work nearly always includes overcoming the absence of trust and connection created in childhood. Remarkably, both AA and the addiction treatment community as an entire realized this fact long before Alexander's rat park experiment. In truth, the usually parallel work of 12-step recovery programs and formalized dependency treatment programs, after the preliminary experience of detox, includes connecting the addict to other people, and not just any people either. We are talking about safe, encouraging, trusted, compassionate people. Much of the time, these safe and supportive people are other addicts in healing who know precisely what it feels like to be addicted and to embark on the prolonged process of healing. Having said that, this newfound sense of connection does not always need to be with other recuperating addicts. For example, as Hari talks about in his TED Talk, the country of Portugal, which legalized illegal compounds, for example addictive drugs, in 2001, takes a broader than normal approach to conquering addiction. Instead of spending money on imprisonment, Portugal has focused its efforts on helping addicts connect with society at large. If, for instance, the addicted was a computer system programmer right before her or his failure, the federal government will find a firm that needs a computer system programmer and offer a share of the addict's income for the first year of work, if the company will hand that person a shot. In addition, the government funds standard treatment opportunities and various other forms of social support. In short, Portugal tries to reintegrate addicts into the human rat park in a way that helps them learn how to trust and connect. And it's working as well. Contrary to most doomsayer forecasts, decriminalization in Portugal did not lead to an increase in substance abuse, dependency, and related problems. Truth be told, the opposite happened. Bothersome drug use is down. Substance abuse amongst adolescents is down. Drug-related deaths and other drug-related harms are down. Jail or prison overcrowding is down, and so on. Having said that, things aren't flawless or perfect by any means. For instance, there are reports that cannabis use is up, and that more people are reporting life use of it. This could be a matter of people equating marijuana with alcohol, and now that it's legal, simply choosing to smoke instead of drink. But in general, it looks like Portugal's unmatched method of linking rather than isolating issued drug users has worked. So it does certainly appear that the reverse of addiction isn't sobriety, it's connection. This being said, developing healthy social connections as a part of recovery and healing is hard. It requires time, effort, and a willing support network. The good news is that we now know for certain that this type of recovery and social connection is possible, even for the most troublesome of addicts. This has been Dopamine, Addiction, Connection, and ADHD Issues. Written by Mark Daly. Narrated by Doug Green. Dopamine, Differences in Gender, Brain Regions, and Tendencies. Written by Mark Daly. Narrated by Doug Green. Chapter 1. What does dopamine actually do? Dopamine is a famous chemical. It holds a treasured position in the annals of well-known science as the reward drug. Countless short articles refer to the latest research studies of food, sex, and exercise as enhancing dopamine and, by implication, enjoyment. However, is that characterization accurate? What does dopamine really do? In the past 20 years, research on the role of dopamine in the brain has continued apace. A key finding is that dopamine is not the benefit chemical, at the very least not in the way that the media would have us believe. In the 1990s, a now classic experiment was carried out by Wolfram Schultz and colleagues. He noted from brain cells that produce dopamine and deliver it to other brain areas. An increase in the shooting of these nerve cells is an indication that more dopamine is being released in the brain. Schultz turned on a light and then delivered a tasty drop of juice to the animal. At first, the dopamine cells responded to the juice, constant with the reward theory. But in time, as the animal started to comprehend that juice always followed lights, the dopamine response went away, though the animals continued to gulp the juice down. Then, when the researchers suddenly stopped the flow of juice, dopamine decreased. This experiment provided a preliminary clue regarding what dopamine was doing in the brain. In parallel, computer scientists were building on early work on trial-and-error learning in psychology. A popular theory, the Rescorla-Wagner rule, proposed that learning should occur when events are unexpected. This makes a lot of sense. If the food at your preferred dining establishment is much as you expected, there is no point in upgrading your viewpoint of the chef. At the time Schultz was doing his experiments, computer researchers were using a version of Rescorla-Wagner learning how to develop advanced computer system programs that could learn, among other things, to play a pretty good game of backgammon. These algorithms count on computing the difference between what you expect and what you get. An error in forecast, or prediction mistake. Revising our expectations using prediction errors is a really efficient way to learn, at least in the long run. Almost every little thing we do in life comes with both a forecast and an associated prediction error signal. The shock we feel getting on a fixed escalator is a manifestation of our forecast that the escalator is usually moving. The Schultz research studies and many subsequent experiments have shown that the dopamine reaction in the brain closely tracks what one would expect from a prediction error signal. No dopamine is released later on in the experiment because the animal has learnt to expect the juice. So, is dopamine a rewarding chemical? In a sense, yeah. What we call rewards are usually things that are all of a sudden good. Consider bumping into a good friend on the street, or getting a pay rise. Those unforeseen events will result in positive prediction errors and increases in dopamine. However, if we get what we expect, even if it's a really enjoyable experience, dopamine probably plays less of a crucial role. Chapter 2. Dopamine and the cerebellum make us really human. Yale-led scientists recently carried out an enormous and detailed relative analysis of human, chimpanzee, and macaque brains. Their paper, Molecular and Cellular Reorganization of Neural Circuits in the Human Lineage, was released November 24, 2017, in the journal Science. This cutting-edge project discovers little but unique evolutionary distinctions related to dopamine production in the neocortex and gene expression in the cerebellum, Latin for little brain, that appear to make the human brain special. To identify distinctions between human and non-human primate brains, Andre M. Souza of Yale University, together with co-lead author Ying Zhu and lots of collaborators from around the world, assessed brain tissue samples from 6 human beings, 5 chimpanzees, and 5 macaque monkeys. During their analysis, the researchers created transcriptional profiles of 247 tissue samples from 16 different brain areas, including the amygdala, cerebellar cortex, cerebral neocortex, hippocampus, striatum, and others. One of the most striking discoveries of this analysis appears to be that people have a lot of cortical circuits in the neocortex and striatum, a brain region most commonly associated with movement that assist in the production of dopamine. As a neurotransmitter, dopamine plays a fundamental role in many aspects of human cognition, fluid movement, reward-motivated behavior, and drives pleasure centers. Dopamine pathways are also connected to a working memory, sensible reasoning, reflective probing behavior, and general intelligence as human beings. Sousa et al. Learned that human brains showed substantial upregulation of a gene, TH, that is associated with dopamine production. Significantly, TH was highly expressed in the human neocortex and striatum, but missing from the neocortex of chimpanzees. Quote, "The neocortical expression of that particular gene was more than likely lost in a genetic forefather and reappeared in the human lineage," Sousa mentioned in a declaration to Yale News. Another unexpected discovery of this research study includes the cerebellum. Evolutionarily, the cerebellum is one of the most ancient regions of the human brain. Historically, the cerebellum was considered by many specialists to be a, quote, non-thinking, prehistoric part of humankind, brains accountable for things like fine-tuning muscle coordination, balance, and smooth or synchronized saccades during eye-tracking movements, vestibulo-ocular reflex. However, recently neuroscientists have discovered that the cerebellum also plays a mystical but considerable role in human cognition, emotional control, and general mental wellness. Additionally, numerous human and animal research studies have found a correlation between irregular structure or functional connection of Purkinje nerve cells in the cerebellum and autism spectrum conditions, ASD. Chapter 3. The Real Results of Dopamine. Just picture the very first taste of sturdy food you ever ate in your whole life. Your dopamine rose and it created the internal feeling of, wow, get me more of this. Let's zoom in on that moment simply because it helps us understand the dopamine urge for more. We're all born with various needs that we can't meet for ourselves. A newborn doesn't even know what milk is. But when you're hungry, your blood glucose falls and that activates cortisol, the tension chemical. Cortisol triggers sobbing, and that brings milk which alleviates the appetite. The nice feeling we get from dopamine is typically released when a real need is met, so milk sets off dopamine in a starving baby. The child doesn't need to understand its needs because the taste of sugar and fat triggers our reward detectors. Therefore, the baby feels good before the milk raises its blood sugar. Our natural benefit detector is quite interesting. Anything that eliminates cortisol is a benefit from your brain's perspective. So if you are passing away of thirst in the desert, water is a big benefit. If you see a sanctuary in the distance, you rejoice and dopamine releases energy that helps you run to it. But in your life today, endless running water doesn't make you happy. You need to actually meet an important need or ease a risk to stimulate dopamine. However, our brain defines needs in a wacky way, which is the reason that we do eccentric things to stimulate dopamine. Neurons connect when dopamine flows, which wires you to turn it on faster when you see something that triggered it before. When a baby hears its mother's steps, dopamine starts flowing. Those dopamine paths help you find a sanctuary the next time you're lost in the desert. But they also help you call the pizza delivery guy when you're in a tiff. Our brain expects benefits where we have experienced them previously, simply because paths build when dopamine streams. When a benefit is more than expected, dopamine surges. A baby's first taste of sturdy food is more gratifying than expected because solid food has more nutrients than liquid. A huge surge of dopamine builds a big neural path, which offers huge inspiration in the future. It's useful to comprehend this from the perspective of our forefathers who started foraging for food in youth. A little monkey is never given solid food. It only has breast milk till it gets something itself. However, Every monkey finds that for themselves, without textbooks or curriculum development professionals, thanks to dopamine. Picture your forefathers' delight when they traveled towards a fruit tree and found an entire grove of ripe fruit. Our brain is constantly forecasting benefits so as to make great choices about where to invest your minimal energy. When you stumble on a reward that exceeds your forecast, that's essential survival information. A rise of dopamine wires it in to help you find rewards in the future. Your forefathers did not get the exact same dopamine rise each time they went back to the amazing fruit grove because now it was already expected. They were still glad to meet their needs, but their fruit needs were soon sated, and they'd get more dopamine by finding protein. Hence, the mission for big benefits helped them survive. Dopamine feels so good that you want to do things that activate it. This leads to a problem in today's world of abundance. Our dopamine circuits inspire us to expect rewards that we never ever appear to get, yet we keep repeating the quest because the dopamine circuit is still there. An easy example is a teenager who wins at gambling. The excess reward builds a pathway that expects to win at betting, which is a flawed guide to long-lasting rewards. All of the behaviors that frustrate us flow from dopamine circuits built by past excess rewards. You can probably think of 10 such behaviors in 10 seconds, with many variations of overeating, overspending, overstimulating, and overanalyzing. It's simple to concentrate on the frustrating circuits of others, but it works to be familiar with your own. You can't go to Paris for the first time each day. So if you expect life to bring you that first-time feeling every moment, you end up dissatisfied. However, you could expect it anyway. Maybe you think other individuals are delighting in a constant flow of delight and excitement. Perhaps you think you'll get it as fast as you reach one promised land or another. When you're disappointed, you choose that something is wrong with the world or with you. The solution is to understand exactly how your brain works. Our brain developed to promote survival, not to make you happy. Dopamine developed to encourage foraging in the world before supermarkets and contraception. Survival required continuous effort, and dopamine made it feel good. Our brain saves the good feeling for steps appropriate to meeting survival needs. Thankfully, there's a way to feel good with this brain we have inherited. Little drips of dopamine are released when you step closer to a reward. You can let small drips be your guide rather than questing for that hit-the-jackpot feeling. Knitting a sweater activates little drips of dopamine with each step closer to a reward. Once you're completed with the sweatshirt, you get a bigger rise when you look at your accomplishment, but you do not expect the surge without taking the steps. Luckily, dopamine makes the steps feel good as long as you see your progress toward the goal. In the long run, you get more dopamine from constant steps than from big rises. However, big surges lure you unless you know how your brain works. Chapter 4. Why is it so difficult to just say no? Why doesn't she or he just stop when she or he knows how much it's hurting me? This is just one of the most common questions asked by the ones that love somebody addicted to substances and/or damaging habits. It may be helpful for those loved ones to know that stopping isn't simply a matter of wanting to, or simply understanding that you could be causing another person's pain. Research shows that to just stop isn't so easy. One of the main reasons is called dopamine. Research on the brain indicates that dependency is all about effective memories, and recovery is a sluggish process in which the influence of those memories is lessened. Both addictive drugs and highly pleasurable or intense experiences, such as a life-or-death adventure, a criminal activity, or an orgasm, set off the release of the brain chemical dopamine, which in turn develops a benefit circuit in the brain. This circuit registers that extreme experience as crucial and creates long-lasting memories of it as a pleasant experience. Dopamine alters the brain on a cellular level, commanding the brain to do it again, which increases the possibility of regression even long after the conduct or drug has stopped. Dopamine also helps to explain why intense experiences can be just as addicting as drugs. Additional research on dependency suggests that dopamine is not just a messenger that dictates what feels good. It also tells the brain what is important and what to pay close attention to so as to survive. And the more powerful the experience is, the stronger the message is to the brain to repeat the activity for survival. In addition, the ones that have less prominent things in their lives that capture their interest and attention are more vulnerable to those things that might give them a rush and alert the brain in a powerful way. This research on dopamine goes a long way in explaining how someone can end up being addicted to something that can become so damaging and harmful in their lives and the lives of those they really love. It usually also helps to explain why meditation, yoga, exercise, and acupuncture can be valuable tools in the battle against dependency, as they resolve the physiology and biochemistry of the individual. Battling dependency isn't simply a matter of willpower, but also is about transforming an individual's body, mind, and life and creating a new set of experiences for the brain to register as important and enjoyable. It is also about patience, healing, not taking regression personally, and the passage of time to allow the memories to fade. Chapter 5. Dreams are affected by dopamine. The role of dopamine in rapid eye movement, dream recall, and dream content has come under progressively more powerful scrutiny over the last few years. This is theoretically crucial for understanding sleep and dreams because dopamine is an effectively characterized neurotransmitter that's central to fundamental inspirational and cognitive activities of humans. Its role in rapid eye movement and dreams was for several years obscure, given the general view that catecholaminergic activity throughout rapid eye movement was reduced. Noradrenergic activity seems to be obviously reduced in rapid eye movement, but the picture is more complicated for dopamine. Early signs that dopaminergic activity in the limbic psychological brain and in the frontal causally central role. For instance, REM invasion in narcolepsy and in schizophrenia seems to be connected with imaginary experiences that could be reversed with antidopamine representatives. Individuals with frontal lobectomies, the frontal lobes are a main forecast site of dopamine fibers, reported lowered dream recall. Mark Solms, in his landmark work on brain lesions and dream recall, supported the association between dream recall and frontal lobes. He proposed that dreaming was, at least in part, a desire satisfaction process, as proposed by Freud, with dopamine fueling the hallucinatory wish fulfillment dream content. He also claimed that dreams occur both within and outside rapid eye movement, which is obviously the case. The recent information on dopaminergic impacts on rapid eye movement, dream recall, and dream content suggests that Solms didn't really need to appeal to NREM dreaming to support the role of dopamine in dreams. Certain animal research studies and research studies in human populations strongly support the idea that dopamine is crucial to REM physiology, dream recall, and dream content. Lampros Paragamveros Paragamverosa and Schwartz present what they call their reward activation model, RAM, of sleep and dreams. The authors incorporate current neurophysiological, neuroimaging, and scientific findings that point to substantial activation of the mesolimbic dopaminergic MLDA benefit system at the time of both NREM, N2 in humans, SWS in rats, and rapid eye movement. With regard to rapid eye movement, dopamine bursting activity within the ventral tegmental area is elevated. This is substantial, simply because this is precisely what occurs in the waking brain when it is processing stimuli that's surprising or of special value. Activity at the time of rapid eye movement in many animals, consisting of, to some degree, in people. Both phasic bursting of dopamine nerve cells in VTA in rapid eye movement and theta activity suggest that REM is processing info of unique value to the organism. However, we really need more information on the role of dopamine in REM in humans. One way to look at dopamine's role in rapid eye movement in people is to study rapid eye movement, dream recall, and content in individuals with modified levels of dopamine in their main nervous systems. De Gennaro and associates have just recently reported De Gennaro L., Lanterio, Pires F., Scarpelli S., Assonia F., Ferrara M., Caltagirone C., Spalletta G., dopaminergic system and dream recall, an MRI study in Parkinson's illness patients. Hum Brain Map 2015, December 24th. DOI 10.1002/HBM.23095. EPUB ahead of print. A fascinating set of associations between indirect measures of dopaminergic activity and dream recall in a group of clients with Parkinson's illness, PD. They studied sleep criteria, dream recall, dream content, and MRI scans of various brain regions in 27 clients with Parkinson's disease, PD, and 27 age-matched controls. Individuals with PD suffer a loss of main dopamine, and the illness is treated with dopamine replacement therapy. Multivariate correlational analyses in PD clients revealed that dopamine agonist dose was associated to lower bizarreness and lower emotional load values in dreams. Visual vividness, VV, of their dream reports positively correlated with volumes of both the amygdala and with density of the left median prefrontal cortex, MPFC. The writers, however, are saying that, quote, association in PD clients between higher dopamine agonist dose and impoverished dream reports, however, and the substantial correlations between VV and mesolimbic areas, however, provide an empirical support to the hypothesis that a dopamine network plays a key role in dream generation. Considering the fact that the amygdala is a highly activated site at the time of REM, it isn't unexpected that intensity of dream reports associated with volume of the amygdala. The relation of agonist dose level with lower psychological values in dreams is counterintuitive for me. Agonists typically stimulate the forward striatum or the nucleus accumbens circuit, which is crucial for emotional processing. Maybe higher dose indicate more efficient processing of psychological info, and because of that, less emotional info in overt dream content? So, yes, the Gennaro et al. Paper adds the growing literature supporting a central role of dopamine in the dream generation process, and a more general role of rapid eye movement in the process of selection and debt consolidation of significant emotional memories. Chapter 6. Dopamine, the left brain, women, and men. Dopamine is the neurotransmitter that is not only responsible for modulating a lot of our physical movement, but also sex, aggression, motivational drive, and, counterintuitively, long-lasting planning and restraint or impulse control. In nearly all right-handed and most left-handed humans who are left brain dominant, dopamine rules the left side of the brain. Lateral dopamine pathways regulate working memory, cognitive moving, and other executive functions such as planning. In the right area of the brain, the artsy part, serotonin and norepinephrine are more dominant. Serotonin and norepinephrine have more to do with psychological activation and arousal systems. The serotonin systems manage our movements and concentrate on a close personal level. They work in close concert with the opiate reward system and some other hormonal and neuronal systems related to close-in work, feeding children, eating, stuff we would do with our look directed downward, and normally doesn't need as much planning. Norepinephrine and serotonin networks also manage our vestibular systems, helping us balance, postural control, and knowing which way is up or down. Again, one associates balance with looking down or being centered in space. Norepinephrine tracts in the brain are also greatly associated with the close-in sense of touch. The dopamine systems in the left region of our brain end up being more active with thinking and planning that's more long-term, scanning the horizon, searching, and recognition. Those systems seldom operate close in our personal space, as it is rare to bring something within arm's reach that we do not already recognize. The median dopamine systems are used in exploration, navigation, and orientation to landmarks. Far vision, upward gaze, hearing, and smell are more connected with the dopamine systems. Dopamine's linkage to distant space appears to have associated itself in humans with distant time also. There is no species rather so future-oriented as Homo sapien. No other species has such high concentrations of dopamine in the brain. An apparent and perhaps questionable correlate to these different neurotransmitter systems responsible for close feeling balance versus far navigation planning is the inherent female-male parallel. Aggressive male behaviors are mediated by dopamine, whereas responsive female and maternal behaviors, grooming, feeding, are moderated by close-in hints, norepinephrine, and opiate and oxytocin systems. It is probably not really a coincidence that dopamine-related conditions such as schizophrenia, addiction, ADHD, and autism are more common in guys, although the serotonin-norepinephrine-connected anxiety and depressive conditions are more typical in women. Of course, dopamine is also associated with depression and opiates with addiction, and guys get depressed and restless while women have ADHD and autism. Those are certainly not absolutes, just patterns. I will speculate here that the differences in personality disorder identifies between men and women might relate to the neurotransmitter dominance differences too. A lot more guys have the analysis of antisocial personality disorder than women, generally a sociopath. An individual with antisocial personality disorder usually does not have empathy for others, and often fights with impulsivity and aggressive drives and drug abuse. That mix will get you sociopathic behavior, taking, murder, and so on. Yet, more women than men are diagnosed with borderline personality disorder. Somebody who has borderline personality disorder typically has trouble with relationships and consisting of feelings appropriately. He or she can have to cope with rage and impulsive conduct also. But typically, the violence is directed at him or herself in suicide efforts or self-injurious conduct. A person with borderline personality disorder has empathy but has trouble applying it. She or he likely has boundary problems and has a hard time sitting with another person's pain without feeling it way too much as her or his own. Therefore, he or she will feel extremely deeply for a person, then when it gets to be way too much, push him or her away. Those with borderline traits also tend to have impulsive behavior and have higher risk for substance abuse. Both borderline and sociopathic people are likely to have experienced abuse or disregard as a kid, but some have not and still have the disorder. Therefore, if we keep up the rather gross simplification that guys are dopamine left brain dominant and women may be more well-balanced between the right and left hemispheres, you can see how troubles with managing dopamine, combined with an underactive social or empathy, serotonin, norepinephrine system, would get you sociopathy in a guy. In a woman, the trouble in borderline character seems to be more in the poor control of both, but perhaps more a deficiency of dopamine and a dysregulation of the social or empathy serotonin or epinephrine side of things. Plus, obviously, there are borderline men and antisocial women. Think Sharon Stone in Standard Impulse. It is a proper supposition that some of our social male or female constructs keep us from wishing to diagnose women with antisocial personalities, and maybe the same social constructs, pressures, and expectations mean that an abused boy child has the risk of growing up into a sociopath, and an abused girl kid has the risk of developing borderline personality disorder. I'm inclined to believe some of that is going on, but that the biology is also at play. Naturally, many mistreated kids grow up without having personality disorders at all. I'll finish up with a discussion of how our brains wind up the way they do in the first place. The same gravimetric serotonin-driven vestibular systems that help us stay well-balanced are responsible for the migration and localization of the dopamine systems as they are formed in early development. The concept goes that our bipedalism keeps us oriented in a different way than other primates or other animals in utero, resulting in different gravity signals and a more lateralized brain development, meaning our right and left brains are just more different than the right and left brains of any other animals. There are no recognized genes that serve as master directors for this lateralization. All of it seems to be associated with epigenetic factors and serotonin accessibility in the right place at the right time. Chapter 7. Does the reward system actually benefit us? Dopamine is trending as the most well-known neurotransmitter. And why not? There are days I think it rules the world, or at least the day-to-day activities of my friends and family. The craving you have when you smell the coffee brewing in the early morning. Thank dopamine. That elation you feel throughout your body when you fall hopelessly and deeply in love. Again, dopamine. Dopamine appears to be all over, giving people a little rush of satisfaction and energy when we need it most. So what's the harm? It's a naturally based chemical that provides energy and inspiration. The harm is best comprehended by recalling the infamous rats in Skinner's box back in the 1950s. Researchers put electrodes into the limbic system, feeling centers of the rat's brain, and sent a little shock to the area when the rat entered a particular corner. The theory was that if the shock was undesirable enough, it would cause the rat to keep away from the corner. Enough shocks, and the rat's brain would wire the corner with the aversive stimuli. Even so, a weird and unanticipated thing took place when the electrode was placed in the nucleus accumbens, a dopamine path that belongs to the limbic system. The rats did just the opposite. Instead of staying away from the corner, they returned to get the shock over and over and over again, up to 700 times an hour. In fact, this was so compelling to the rats that they opted for the stimulation over food. The rats could not define craving to us, but certainly the recurring nature of their dopamine seeking made it clear that this was something they needed to do. The boost in inspiration and energy that dopamine offers can be a good thing, but when your brain gets wired to compulsive habits that stimulate the dopamine benefit path, addictions, then your life can be as out of control as the poor rat in Skinner's box. So dopamine itself is not the problem, nor is the dopamine benefit system. Dopamine is simply the carrot on a stick created to give a reward to life-sustaining activities like eating healthy food, having sex, drinking water, and being held in supporting relationships, so that you're going to keep doing these healthy things over and over again. The problem is how we stimulate the dopamine path. In an ideal world, one that comprehends the midpoint of healthy relationship to health and wellness, the dopamine reward system remains connected to human connection as the main source of stimulation. Unfortunately, we don't live in this ideal world. We live in a society that actively undermines this valuable dopamine relationship connection. We raise kids to stand on their own 2 feet, while the separate self is an American icon of maturity. It's making us sick. This detachment is a setup for dependency as we search for other sources of dopamine. The other sources look shockingly similar to the list of common cultural grievances: overindulging and obesity, alcohol and drug abuse, consumerism, persistent connecting. Not only do these addicting, harmful behaviors get paired to the dopamine benefit system, but they produce a feedback loop of seclusion that pushes people to more addictions. With no healthy relationships, we all end up like the rats in Skinner's box, seeking dopamine from all the wrong places. It is time to rewire our brains for the healthy relationships and connections that reward us with positive energy and inspiration. Chapter 8: Dopamine and Iron. Iron, a typical mineral and, paradoxically, a common mineral shortage in humans, is very important to the brain and important for normal brain development and human behavior. It is a key cofactor in the making of neurotransmitters, substances in the brain that influence nerve cell signaling, including but not limited to serotonin, norepinephrine, and particularly dopamine. Iron is also necessary for the enzyme monoamine oxidase to break down these neurotransmitters. Imbalances in levels of the neurotransmitter dopamine can result in muscle and behavioral problems. Depending on the location and direction, high or low, of the imbalance in the brain, dopamine issues can cause tightness in muscles, tremor, irrepressible urges to move while at rest, also known as agitated leg syndrome, issues with attention, concentration, inspiration, and depression, but also psychosis, fear, and aggression. Restless leg syndrome is usually successfully treated with iron supplements, and the numbers of dopamine receptors in areas of the brain will change depending upon the iron levels in the brain. Attention deficit hyperactivity disorder is a typical condition occurring in 5 to 15% of kids and up to 5% of adults worldwide. It's typically hereditary, and as a result of the common symptoms of motor restlessness, poor concentration, and distractibility, it is thought at the very least in part to be caused by problems with dopamine levels, or the efficiency of dopamine receptors in the brain. Medications that help symptoms of ADHD, such as stimulants, regulate the levels of dopamine and norepinephrine in the brain. Methylphenidate, trade name Ritalin, for instance, is a dopamine reuptake inhibitor that keeps more dopamine in the synapse. In genetic studies, certain dopamine transporter genes and D4 receptor genes were both connected with scientific signs of ADHD. Several research studies have now revealed that ADHD in children is associated with low levels of serum iron and ferritin, another procedure of iron, compared to control kids without ADHD. Iron supplements have also been shown to enhance symptoms of ADHD. Low iron levels have different sequelae in children than in grown-ups, specifically when it comes to behavior and development. Iron shortage in young children can lead to permanently lower IQ, growth-related delays, and behavioral disturbances, which can only be partly ameliorated with iron supplements later on in youth. Neural development begins before birth, of course, but dopamine neuron maturation and migration continues through adolescence, with subsequent adjustment and pruning happening up until adulthood. Iron levels influence neuron development, shown in animal studies. It's possible higher occurrence of iron shortage in children than grown-ups, along with the slow progress of dopamine nerve cell maturation and development, could partially clarify why children tend to have a higher incidence of ADHD than grown-ups, and the signs in children tend to include more motor uneasiness. Following this line of thinking, early recognition of iron shortage and iron supplementation in children may ameliorate and even prevent some kinds of ADHD by positively affecting dopamine nerve cell development. There are limited research studies of grown-ups with ADHD, and the correlations between iron shortage and symptoms aren't so clear. Apart from this, minerals and vitamin supplements have had combined results as treatment. In these populations, though, the only typical shortage found was vitamin D. More research needs to be done to totally comprehend the connection between iron, dopamine, and ADHD, but it would make a lot of sense for folks with ADHD signs, especially kids, to get serum ferritin levels measured, and to loaded iron deficiency when found, since iron excess can also trigger illness, and kids with their smaller body sizes are more susceptible to the effects of minerals and vitamin overdoses. It wouldn't be safe to aggressively supplement iron without checking levels first. Chapter 9. Why Goals Don't Bring Complete Satisfaction. We're awkward with dopamine dips for good reason, obviously. Dissatisfied chemicals command your brain's attention the moment happy chemicals droop. Things could seem terrible suddenly, even if they're not. Dissatisfied chemicals develop to inform you to survival threats. They feel bad since that gets your attention. At times, we can relieve unhappy chemicals by fixing the underlying issue, like eating when you're hungry or sleeping when you're tired. However, some dissatisfied substances will always be there to remind you that life is limited and you're not the one in charge of the world. You can disguise your unhappy substances by doing things that triggered your happy substances in the past, but that will only work for a short time. Happy chemicals did not develop to rise all of the time. Their job is to get your attention when something promotes your survival. They turn off soon after they turn on, so they are prepared to get your attention to the next good idea. A recent monkey study makes the ups and downs of dopamine remarkably clear. Academics trained a group of monkeys to do a small job in exchange for a spinach leaf. Then the experimenters rewarded the monkeys with squirts of juice instead of spinach. Juice is a lot more satisfying than spinach, simply because it has much higher energy value. The animal's dopamine skyrocketed. Dopamine is the brain's way of saying, "This really meets your survival needs." Then something curious took place. The monkeys' dopamine fell over time. They continued getting the juice reward for the job every day, but their brains stopped responding to it. This shows that dopamine is the brain's reaction to new information about new benefits. Once the juice belonged to the routine, no effort was needed to get it, and no dopamine was needed to make a record of the survival lesson. This experiment has a remarkable finale. The experimenters stopped the juice and switched back to spinach. The monkeys responded to the spinach with fits of rage. They had come to expect the juice. They were obviously unhappy when they didn't get it, but it did not make them happy when they had it. This is the survival mechanism we've inherited. Old benefits do not make us happy, because the brain soon habituates to them. It takes what you have for granted and focuses its attention on new benefits. If you could grow and better benefits in every moment, you would never ever have to experience the core unhappiness of being a mortal human being. But that desperate looking for causes distress of its own. This distress is typically blamed on our society because people do not understand how they are creating it in their own brain. You are free to step off the hedonic treadmill anytime you choose. You can do it in one instant just by accepting your dissatisfied substances rather than hurrying to mask them with happy chemicals. You'll find that your unhappy substances are not nearly as horrible as the routine of running away from them. Rather than getting annoyed with your neurochemical ups and downs, you can thank your brain for trying to promote your survival. This brain we have acquired from earlier mammals has successfully promoted survival for so long. A hungry lion spurts dopamine when it finds a victim. A thirsty elephant releases dopamine when it finds water. A monkey's dopamine flows when he nears a juicy fig after climbing a high tree. Dopamine kept our ancestors going on long efforts, whether stalking game or storing grain for winter. Dopamine tells your body to release energy reserves just because the objective is near. This has been Dopamine, Differences in Gender, Brain Regions, and Tendencies. This has been Dopamine, the function, meaning, and side effects of neurotransmitters.